We have Samuel. Can you hear me?
Yes, I can. Sorry, I was finding my... Hello. Nice to
How are
meet
you?
you. Thank
Nice
you
to
for
meet
having
you.
me here, Jerome.
Oh, you're welcome. We used the first few minutes to introduce people to the group. So why don't you introduce yourself and we see how we can help you.
Yes, I don't know the answer to the first one, the second point yet, but I can answer the first bit. My name is Sam Solomon. I'm a physiologist by training, I suppose. I've been interested in adaptation and related processes for many years. I've collaborated fairly closely with Sarah Rudiger over the last year or so on some related questions, trying to develop some related questions, and she had told me about what you're doing here, and I thought it was really interesting. At the moment, I'd really like just to sit back and see if I can understand what you're trying to do rather than to try and ask you to give me something, but and just to work from there if that's possible.
No worries. Fine. Welcome, Samuel. It's just it's a good way to get people to talk to each other at the beginning. Our process is fairly I think we are learning as we go because those kind of community projects are new to me, I think to many people. So we are trying to sort of exploring the space as we are sort of doing the analysis at the moment. Okay, should we get started? I feel like we have quorum. Okay, so good morning, good afternoon, everyone. So our agenda, so first, this meeting is going to be recorded and posted on YouTube so that people can follow asynchronously. I remind everyone that if anything occurred during the meeting that you want us to edit out, we can crop. So just reach out at the end so that we can remove that content. Our agenda for today is to have sort of an unstructured discussion around analysis that people have prepared. Last week, I asked for people to post the content of 2 slides on the discussion thread. I've seen four groups of analysis that were shared. The first one is from Ido. The second one is from Roberto, Sarah Rudiger and their group. And we also have this is still Sarah, and we still have something from David. So I feel like that's 4 things to talk about. That's plenty. Does that sound good? I propose we just follow from the top to the bottom in arrival. Does that make sense for everyone? Ido, can you start? So let's try to keep each person between 10 to 15 minutes so that everyone has the time to speak. So I would advise just to have like a 5-10 minute discussion of your content, introduction of your content, and then 5 minutes discussion from the group. You want to start, Ido? Do you want to share your screen or do you want us to go through the discussion forum?
You can go over it. It's fine. Yeah.
Okay.
So first I want to say that I'm not an experimentalist, and it's like the first time that I'm working with real data, because usually I'm doing simulations and training deep neural networks. So this is like kind of my way to go into more data-driven stuff. So I don't know if it makes sense. Please correct me if it's wrong.
What you're doing makes a lot of sense.
Okay, great. So I'm mostly interested in the SLAP2 data, and specifically with data that is related to both glutamate and calcium. But apparently there is not such data now. So like as a first step, I saw that one of the figures of the paper is about the quality control of the data. And I saw that the panel for the figure for SLAP2 was missing, so I tried to do that. So I followed the structure of the figure from the MIST thing. And basically, this is only analysis of the glutamate data from SLAP2. So the first panel, I think it's panel C, just like showing the different recordings, like different planes for the proximal dendrites and the apical dendrites, and the ROIs that were provided in the experiments. And already the ROIs are classified to three groups, which are low SNR, intermediate, and high SNR. And in the next panel, it will explain how it's working. And the last thing in panel C is basically one ROI. It shows like the row trace and the detected events. So in the beginning, I did something very different from this, but I saw that in the mesoscope data, there was a meeting about it, and they tried to basically detect spike events. And I thought here it's relevant to do something similar, but to detect putative glutamate release events, basically. So just like try to classify the events and how significant each event is. So it's very simple. In G, it just shows that basically we measure the like the noise level of the signal, which is basically the mean absolute deviation. And then each peak, basically we can classify it by its standard deviation from the noise flow. And if it's like above 4 standard deviation, then it's more significant than 2 or 3, for example. And then for each synapse, so it has many events basically, and we want to classify each synapse to say, is it can we trust it? Is it low SNR or high SNR? So this is basically a k-mean clustering on the data, which is two-dimensional. One axis is the number of the percent of events which are less than 2 standard deviations, and the y-axis is the number of events which is larger than 4 standard deviations. So basically, if it's in the top left, it's a high SNR. If it is in the bottom right, it's low SNR. So basically, there's like nice separation. And I don't know why, but it seems like that there was some sessions with glutamate only and some sessions with glutamate and calcium, and it seems like that the one with glutamate and calcium basically are much better usually. So it's all more to the top left. And the last panel, which is K, it's basically to try to classify these 3 classes across different contexts, across different like plane of recording, which is like a proximal or apical, and across time. So I think like the main thing maybe it's that the recording gets better over time. I have no idea why, but it's like the recordings which are like more recent, they have more synapses which are considered the highest NR. And another thing that I thought that it should be is that the proximal dendrites should have more synapses which are higher SNR, because it's maybe closer to the, maybe, I'm actually not sure, but
closer to the surface, yeah.
So is the proximal more close to the surface or the apical?
The apical.
Okay, so the apical should be higher SNR.
In principle.
Okay, so it seems like in the glutamate only session, like in the bottom left, it's a little more like that, but in the glutamate and calcium, it's the other way around.
Yeah, ideally we are not too sensitive to depth because we're I think 90 microns away, right, Mehdi?
Yeah, something like that. I don't think there will be a huge difference. Maybe it's more about the tuning of these synapses, like how tuned to the presented stimuli they are, so how much glutamate release there is, that then we will detect more fluorescent based off of it. So that's why maybe it changes from session to session.
Okay.
I really like your analysis. I think it's exactly what we had in mind. I feel like you really touch on it. I'm just curious about the metric you use and whether because Madi is in the process of writing another paper on the microscope. So can you tell us, Madi, what you think here about this metric?
I think something I'm not sure is, so we are trying to match like mesoscope QC analysis, because for example for this lab 2 paper we just looked at the distribution of SNR. We computed amplitude SNR, just we found the noise floor and I don't know, like 95 percentile of peaks, and then like that peak to the noise floor is the SNR, like so the definition of SNR that we use, and just we looked at a distribution of that, and then we looked at example traces from different places on that distribution, and just to get a feel of how different synapses with different SNR values look like, both in their single trial activity and also trial average, like stimulus triggered average activity, just to get a feel of what does SNR of 12 mean. But that's all we did to characterize SNR. So here, if you want to, yeah, go ahead.
How did you estimate the noise floor, Madi?
It's the standard deviation. It has a name, this method. So you look at changes across a consecutive. So you differentiate the activity. So you look at like fluctuations, and then you look at the standard deviation of that fluctuation.
So is STD or the DF?
Yes.
Okay. Would you be willing to share some of us analysis with Ido? Or?
Yeah, of course. Yeah, there's a function that we wrote. It's like a very simple one, actually. And the reason we picked this one is that it's so simple. You just have a feeling of it. But again, it just produces values, like SNR is like 10, 7, 12. Okay, what does it mean? That's why we provide example of those traces just to calibrate. But yeah, I can just share the function. It's so easy. Yeah.
I think the finding that these is really interesting. I think the instruments got better over time. I think that would be expected because we got better. It's a very new effort. And would you agree, Matt, if it makes sense?
It's possible that our expressions are better. Yeah, we figured out the best, like better, tighter. So it's possible that the expression is better. Yeah, maybe the microscope has been maintained and improved. Also, how you choose the ROIs to image, that has huge impact on the SNR. So with SLAP 2, you choose what you want to image. And if you stack so many ROIs, then the imaging rate drops and it's not uniform, the drop. And then it can give you very different SNR. Like some synapses will be very low SNR, some synapses will have higher SNR. So like how uniform you sample the region of interest, it will also have a huge impact. And I think over time we got better at like how to choose those RIs to have a better and more uniform sampling. It could, so I think there are like multiple factors contributing here, to be honest.
I'm curious if others in the group that I looked at SLAP2 that I have some comments for Ido. I mean, as far as I see, this is a great analysis to include in the data release paper in the SNR for SLAP2. And so I would propose that Ido and Made iterate on the metrics to make sure that we agree. And then once you have that, we just regenerate the figures with whatever metrics you like, and we can include it into the data release. Does that make sense to everyone? Or?
Yeah, sounds good.
Yeah, sounds good to me.
Okay, Ido, do you have things more things you want to discuss? Or I feel like thank you so much for the work. It's really high quality.
Sure, thank you. There were two things. So one is that I defined the SNR differently for the glutamate only sessions and the glutamate and calcium sessions. So maybe I'm not sure like it's the correct way.
It should be the same. Why was it different?
Because I saw that in the glutamate and calcium, it's like much better. So I wanted to separate it even more. But yeah, maybe it should be the same. And also Alex Meyer, I think, he wrote in the discussion about this that maybe we should actually, it's like it could be a continuous measure. Separating it into like 3 classes is kind of arbitrary actually, and so I'm not sure what is most useful, because it could be actually like some kind of continuous SNR thing. So for example, the ROIs can be like colored from something to something. So if you, for example, go into the discussion, I think it's 172, then there is like the same plots, but all the synapses are colored. So
yeah. But I think the intent of that classification, as I was thinking about it, was to highlight. So this is a measure of the distribution, right? And seeing over the days, over the sessions, or how that distribution changes is important. And so I thought this was helpful, even if it's continuous. But Sarah, what do you think?
Yeah, I'm just wondering, I'll show later basically this continuous way of displaying the SNR for the electrophysiological data. And maybe eventually if we look at the different analyses, we can choose a common plot that we like for all modalities. So we have a kind of, standardized plot, but I think either way, whether it's continuous or discrete, if we find a common way that works for all data sets, that probably would be most useful for a reader, basically.
I agree. But I mean, I guess what I would advise here is that either if you could make a pull request to the data release paper with your analysis and the code to replicate the figure. Once we have the code for all modalities, I think I'm going to merge them into one. I think that'll be easier if I do that, and then everybody can wait on the merge figure.
sounds good.
Yeah.
Should we proceed with the next set of analysis? Oh, okay. Thanks, Ito. That was really helpful. Okay. Next is Roberto here. Yeah.
Yeah, hi.
Okay, Roberto. Do you want to speak to your analysis?
Yes, should I share the screen because maybe it's easier to go
Yeah,
through?
whatever is simpler for you.
Okay. Right. Okay, so yeah, this is really simple. I started by just plotting PSTHs, like also David will show later, I think. But I focused really on one, so only of course on neuropixels sessions, and only on the sensory motor stimuli. Because already in this context, we have actually 3 different sort of like unexpected stimuli. That is the stop of the grating, then we have the omission of the grating, and then we have a change of orientation. And so I just wanted to plot the responses for each session to these 3 different types of oddballs. So you see here in purple is the orientation, in blue the omission, and then you have the alt in red. And we have isocortex, hippocampus, thalamus. I mean, these are the 3 biggest ones because really usually we have a few neurons in the striatum. And so the first thing I notice is that there is a big difference in the responses. Like the orientation one is much stronger than the other 2. And the alt one is actually almost always kind of silent. But we can just scroll through quickly. Oh my God. Like for example, you see this animal that is part of the motor cohort. So we also have this difference, right, between animal trained on the feedback to the running and animal that were not trained. And this is one of the one that had the training, and he also has this really nice like response to the alt. But if we go through, you see that this doesn't. This doesn't. Oh, okay. I can see it like this. You see that most of them, they really don't. And there is usually a small response in the cortex to also the omission, but is much smaller than, in this case, we don't have the histology. Okay, I'm going to scroll through. This is the second one that actually has a big response to the motor alt, but it's really only 2 of them. Okay,
Roberto,
yes.
have you, because some mice will walk and some will stay steady. Did you do any sorting here?
No, this is, so here I'm just looking at, so good units according to the usual QC filtering that is done. So presence ratio, ISI valuation, and what is the third one?
No, I'm just saying when the mouse is walking on the disc as the oddball is inserted, the effect of the alt is much more perceivable. Because
compared to, because it's moving
because the mouse is controlling the screen, and so you would expect context sensitive oddballs here.
So you would expect the blue, the red one to be. We should like divide between running and standing. Yes. Yeah, of course, that makes. Somehow I thought that this was happening only during running.
No, so it's all the time.
Not. It's I see. Okay, yeah, then I should definitely divide this because maybe it will be stronger, yeah. And okay, so then I just quantified this. So I take the average firing rate, and and this is the quantification. So for the alt, you see that we have only 2 that show like an increase in firing rate. So this is a 1 spike per second average increase in firing rate. Here I'm not even zeta scoring. This is just the raw firing rate. And you can see that omission that at least is a bit of a bigger response, but the orientation one is much bigger. And what is cool is that we can split by the cohort, so the sequence versus motor cohort. And you see that actually also the omission, the animal trained on the motor feedback, they actually show a bigger response. And and then you can go deeper here. Oh yes, there is
Samuel?
Sorry, I was just curious, is the stimulus, is it drifting when it's placed on there, or is it static?
So these are all drifting, right?
So the major difference between, there's a major difference between these 3 conditions in that for the emission and for the halt, the stimulus is static, but for the orientation, the stimulus is renewed over time, right?
Let me explain this design a little bit. So here you have a mouse during loose block that is walking on the disc, and the flow forward back of the drifting gratings follow the disc speed. So if the mouse runs really fast, it goes really fast backward. If it doesn't move, it's steady. And then we introduce during loose period different types of oddballs. So one is alt, so the drifting grating just stopped being coupled to the disc. Another one is an omission where it turns gray for 250 milliseconds, I think. And 2 other ones where the orientation just now changed instead of being vertical. So this
yes, I understand that. I think my point is that for the conditions where it's a halt and an emission, the stimulus really is a very brief transient change in contrast, which is then static. So a gray screen or something that's static. So to compare between the different conditions, would you not need to just look at the initial response rather than the sustained response?
You have to compare with the control block, yes, which are separate from this, where we measure the response to the, Stefan?
Do you happen to have some videos of these movies? Because I think would be quite interesting to see, especially because if you've got something which is going to be so vertical in the direction of movement of the grating, is going to have a weird alias with running, right? So theoretically, if you've got vertical bars which move and you run and that moves it as well, right? You're actually going to sum the direction of movement, and it's not at all clear as to what would be the perception of that. Would be nice at some point to show some of these videos. I understand what you're saying for the design, but the combination of the two is not always intuitive.
That's
And
right.
I also agree that the analyses would be quite important to be done as a matter of time, because quite a lot of the surprise might not be equal as a function of time.
Yeah, we have a movie on the data release paper that illustrate a couple of those events, and you can watch it. I can definitely improve the viewer there, but I agree with the controls. Tim, you have a question, Tim?
Cannot hear you.
We can't. I thought I heard you, but I'm not sure.
He says he doesn't have a mic, but it'd be nice to have the code repo for these discuss figures to work with.
Yeah. So I think, Roberto, you could commit the code on the P3 data release paper. What do you think? Or you
Yeah,
want to
sure.
have?
Sure, yeah.
I feel like this needs a little more work. Would you agree?
Yeah. I mean, this was just to explore the data a bit to understand where to focus. You know, I didn't.
Yeah.
Absolutely. Yeah.
I feel
And
like
just
comparing, checking the context of those oddballs might give a very different answer, and then you might have to compare with the control blocks to see if the response is bigger or smaller than outside of its context.
Sure. Yeah. And anyway, because I was expecting, a big response to the motor alt. That was I was a bit surprised that actually
Yeah.
it's quite is only like in 2 out of yeah. And
Yeah, me too. Let's dive deeper.
Yes, and then you can go and check by neuron type, and you also see, so you can go by layer and then check differences by layer, and then you can check by neuron type too. Like here, for example, just SSTs. It's quite interesting that, so here in blue it's layer 2-3, and for the omission and orientation, actually also here it looks like there is an increase. But yeah, this needs more work, definitely. And this is just in the HTML file that was shared, so everybody can use it. But I will also share the code, of course. Okay, but that's it.
Yeah, I think by layer will be interesting. I think if I recollect Georg Keller's group, whose units are more superficial.
Okay, so that would make sense then. Because this appears really only, like if I put all together, I don't see a difference in SSTs. But if I focus only on layer 2-3, I do see a difference. Like where the motor cohort has a stronger response than the other one. Yep. Okay.
There are more questions from Sarah?
Yes.
More quick comment. So if you look at the original paper from Georg Keller, which was imaging based, you will also see it's actually a subset of cells. And so I think what you find is, maybe diluted by having lots of cells which are not modulated, which is not something that's wrong. It's just that you might bias the effects by lots of cells which are not modulated. So I would honestly love to see this data broken down into individual units, like, almost like a scatter plot of each unit's activity during the standard condition versus the oddball, to get a feeling for what's the fraction of cells that's changed and how strongly are they impacted by the mismatch.
We also have imaging data.
Yeah, I know, but in terms of IFIS, right, it's equally interesting, I think.
Yeah. I think there was a more, I mean, it looks like Carter, do you still have your hand raised?
Yeah, I was just wondering, Roberto, where did you get the SST labels in the file?
No, it's not in the file. I computed them. Yeah, it's like from this. I did the one pull request, right, with this SST heat map, and from there I got the positive ones. Yeah.
Okay, I got it.
Yeah.
Do you think that code is ready to be shared? I think you shared it already, Roberto.
For the SST's identification.
Yeah.
Yeah, that's already there.
Okay.
Yeah.
Okay. Yeah, that could be merged into the paper already. Yeah, I think you made a figure already.
Yeah, there is already there is a figure associated to the optotyping. Yeah.
Okay. Do you have more to share, Roberto, or do you have the feedback?
No. It was just these two things, really. Yeah.
Yeah, thank you. This is great. I feel like it needs more investigation.
Agree. Sure.
Should we move on to, I think is it Sarah now, right? This is Sarah.
I think David wanted to go next because he also has an interactive figure from his comment in the chat.
Okay. Do you want to go, Sarah? David?
Yeah. It could be quick, so there's time for everyone. Yeah, so I kind of did something similar to Roberto, but instead I also plotted the heat map of the PSDH across neurons, and I focus only on the neuropixels because I quickly realized that making a figure, across different modalities is just going to be too messy. But in this interactive figure, you can select the different types of experiments that were done. So the oddball, the sensory motor sequence, and then the duration. And then I basically, for each of the different contexts, there's different mismatch events. And from there, I basically focused on, plotting things that I would plot. So, first plotting just the raw firing rate for the mismatch and the control, and then plotting like a z-scored firing rate that's defined on some baseline. So usually pre-stimulus. And then actually maybe it makes more sense to use the oddball first because this is to me, the most simple stimuli set. And then basically like sorting things so that it's, you're looking at what you want to look at. So this is one experiment and there's, 1,300 neurons that passed QC. But we don't want to look at all 1,300 neurons. Maybe we want to just look at the, the visual areas. And maybe we don't even want to look at the multi-unit activity. So we click that out. So there's really only 200 single unit neurons. And I like to plot things, sort things differently. So you can plot things like based on the depth, time to first peak, or response magnitude, or this thing called raster map, which nicely, clusters neurons based on local and global properties. And then basically you have the mismatch actual, context block, but then you also have the control block, which you can plot in the same order, the same sorting as the mismatch. So you can kind of like by eye see in this context the increase in firing rate for the mismatch event. And then this is a plot that's very similar to Roberto's in that this is the average across all neurons in the visual cortex for the context signal, and then also the control signal, and it's the dashed. And I also took the SST code from Roberto's repository, so you can also look at SST cells, of which there's 7 in the visual cortex. So this is just for one animal. I didn't do it for all animals because the way that the backend is set up for the interactive figures based on Jerome's code, I think based on what I was asking the AI agent, like it might be prohibitive to do every single animal. I don't know. Because basically it's saving like this, this matrix per, I guess, event. And obviously there's 4 different stimuli sets here. So that's kind of like the interactive figure. The static figure, I feel like it created something that like I don't really like, but it's basically trying to quantify the relative increase in firing in the mismatch event minus the control. But this is averaging over way too many things. So this is not a useful plot, I don't think. But seeing Roberto's plot made me think it'd be maybe nice, we could maybe combine some elements of both figures if we wanted to actually quantify oddball responses. But I thought that for a data release paper, like looking at the signals by themselves and not really quantifying like, what an oddball response is. Because like, look, there's an initial response here, but it's different, right? For different types of, so like motor halt for this one animal doesn't really elicit any, mismatch response. So yeah, that's my interactive figure that Claude helped me make.
I feel like this is really intuitive. I really like the layout because you can navigate. What do other things?
I think it makes sense that you like it because it's taking elements from your interactive figures, right? So, but yeah, I mean, it's amazing that like I was like having this conversation. I'm like, I want these things. I mean, obviously, I had to iterate like a lot. Actually, sometimes it would make really dumb decisions. So like actually defining the baselines properly, you had to be careful about. So like it would just like assume, oh, I'm going to use 2 seconds prior to the stimulus as a baseline, which would then you're averaging over the previous stimulus event, right? So here, like the baseline is only the previous, whatever it is, 384 milliseconds. Whereas the duration, like you have to tell it what to use. So like the 100 millisecond delay, I was actually using a baseline that's 2 trials back. I don't know. Maybe it's not that important, but like I think that being careful with like some of the assumptions that the AI is making, especially when you're doing some sort of computation, is like, yeah.
Well, what I like about it is that we could introduce this as, I think it's figure 7, as a way to teach people how to first extract the oddball response. So you just say, look, if you look at the raw data, it's scattered around, you see the effect of z-scoring and how it sort of pull out, and then see how you can compare with the control. I feel like in the text, that will go really smoothly with that kind of figure.
Yeah. Anyway,
what do other things?
Yeah, I like it too. I think we can join it in a way, like probably like some. I think it's a good idea.
Yeah.
So Roberto uses a different backend than the way that you set it up, Jerome. But like, I don't know. Yeah, there are different aspects to each of them that are nice. Like I think that the Marimo is nice because you can share these HTMLs really easily, whereas like this is like you have to like it's like I think it's like creating it every single time. Like I don't know how it works on the back end, but
yeah, that makes sense. But I feel like the message of the figure is didactic explanation on how to extract the oddball response, which I think will fit very well in the goal of the manuscript.
Yeah, because I think that there's so many ways to quantify oddball responses that like if we just do it for people, it's like kind of like I mean maybe we should still, but like yeah, there's a lot of things you can choose to do.
So you're saying we shouldn't quantify too much. We should just is that correct?
That would be my but
Sarah?
Yeah, I kind of agree with David because I mean, if I look at the layout for figure 7, it's all about understanding the different modalities and what the data looks like, and then kind of helping the person choose which data set and which sessions they want to work with, right? And then within oddball is another whole beast, right? In which parameter space you want and which conditions. But I think the first bottleneck is just trying to understand what the data is about. And so I like this figure a lot. I think it's super helpful. It also helps realize the complexities of the analysis, right, in a way. But I don't think we have to quantify too much of the actual oddball responses themselves.
I mean, Jerome,
Maybe as an example, maybe some example neurons would be great, right? To show that there are indeed oddball sensitive cells in this data. That would be maybe comforting to some.
yeah, choosing responsive cells before you quantify the oddball response across all cells could be one way of maybe strengthening the message. But as Jerome correctly pointed out, like the behavior of the animals is very different across the experiments, and I don't plot anything about the behavior of this animal, but it could be nice to have like the behavior below that shows like the average running speed on a given type of trial. But
or just allow to navigate, yeah, that groups of trials.
Yeah.
Okay. Well, do you have a PR already, or are you planning to submit one, or?
Yeah, so I also mentioned in the discussion that like I had it different, like I have a branch and I can submit a pull request. It's not a fork or anything, so I guess it's the same though.
I feel like we should merge it and have people play with it so that we can provide inputs and iterate it together.
But Jerome, correct me if I'm wrong. In the data release repo, these visualizations require like pulling snapshots of the data, right?
That's what David did. I think he pre-computed some things. So it's going to have to be committed to the repo, which is fine.
Yeah.
Okay. Should we move on to the next? Sarah, you want to
Yeah, sure.
share your screen, or you want me to
Let me share my screen. I just need to find the right presentation. Yeah, okay. Yeah, so basically what I'm presenting is kind of on behalf of Mariana and Yuheng. Mariana's traveling today and Yuheng is asleep. And so, but they basically both took a step back to try to get that really the basic metrics in terms of electrophysiological data that I would want to see if I choose how to analyze this data. And so the first thing we looked at is basically just the signal to noise distributions in this case now for units which were classified as good based on, you know, established metrics from previous publications, and plotted these as a function of the different probes and the different experimental cohorts. And so I think what you can take away from this is that it's basically up to the, you know, person who wants to analyze this data to choose a signal to noise that they want to live with in terms of units they want to consider. But typically, a lot of people use 3 or 4, which in all of these instances would be probably really the good units in that sense that they have a reliable signal to noise. And then we were wondering, like, how well do these, you know, preps work over time, because these are acute insertions, and we were worried that over time the prep might degrade a bit. And so in panel B, we basically looked at the very first recording session for each animal versus the very last, which is basically blue is the first and last is in orange. And what you see is overall, a lot of the experiments are remarkably stable. But if anything, it's true that the last session has a shift to the left in terms of the distribution of the signal to noise. So they get a little worse over time. But overall, I mean, some of the experiments are really remarkably stable in terms of the quality. And so then next we were wondering how reliable are these probes inserted as a function of depth. And so we basically just plotted kind of the firing rates for each individual and each insertion site and color-coded the firing rate as a heat map, which kind of gives you a sense that, I mean, probe A has something at the tip of the probe which has a much higher firing rate. And then in other probes, you see that sometimes there's gaps between zones where you probably cross different boundaries. Yes, Severine?
Hi, I wanted to know, did you plot all the cells, all the output from the sorting, the spite sorting, or did you plot just the good units?
Only the good units in this case.
Ah,
Yeah.
okay.
We could go back to multi-unit, but we were trying to just be agnostic and for now see, yeah, where the good units located. But in terms of, yeah, mapping regions, we could also just take multi-unit overall, basically. But the idea was, it gives us a little bit a sense for the reliability of the probes. And I have to say, I mean, you're remarkably good because the bin size here is like 100 micrometer depth. And I mean, some of these look like they're aligned from the get-go, right? So you can refine this now with your histology reconstruction or with current source density analysis and so on. But I mean, you're very reliably from the get-go. I mean, for instance, the sequence cohort, if you look at the panel for probe D, I don't know, but maybe I should zoom in. You can literally see even like the surface of the brain starts almost at the same place for every individual, right? Because I mean the insertion is so stereotypic that you almost always get exactly the same transition phases from into the brain at the same channel position, which is remarkable to me.
But Sarah, I think there's something weird about like the depth here because we don't have too much insertion that goes beyond 3,000. So that's why I don't understand what are the units between 3,000 and 4,000, for example. But that would be an easy check when you get the CCF, because you have like all the channels with like labeled by area, and the one that are outside are called void. So you could completely check like which channel starts by void, basically, which is the channel, the top channel. So
that sounds great. Yeah, I think you're right. It's like 3,500 or so is the boundary, and then some of these upper events might be noise. We had also this other concern about units with negative depths that we didn't fully understand at some point. And
so I think. Oh, sorry. Go for it.
It's just the boundary conditions we need to check very carefully, I think.
So what we did yesterday is that exactly what I just mentioned. We went back to the CCF and looked at the units that were outside a negative, basically, depth. And then we found them all being noise, basically. So that's why, again, like I'm not sure I understand. If you plotted only the SUA, then I don't understand.
Let me double check. I have to double check with the other two guys. Maybe there was some criteria missed so that we ended up still including some noise. But yeah, I have to check. I honestly don't know at this stage. Yeah.
Thank you.
And then, basically Mariana then proceeded from Yuheng's work from this, and she was curious about the different cell types we would get, because we have basically probes right across many different parts of the brain, not just visual cortex. And so she used basically wave map to classify the different waveform shapes. And what she has done here, she basically tried to color code with a kind of rainbow spectrum the different classes of cell types that she's getting, to then use these to visualize the properties of different classes in the next slide. And I mean, we can discuss, right, if this is an overkill in terms of the number of classes, but because we had prefrontal cortex, we have striatum, we have thalamus, we were basically just trying to be very generous in terms of the number of classes we would be happy to identify at this stage. And then from this, basically what Mariana did is she tried to visualize the different properties. So the plots on the left are not really ideal yet, but in principle, the idea is that we would visualize different properties that I would care for as an electrophysiologist. I would want to know the firing rate, the amplitude, actually both metrics also over time of a session, signal to noise in general. And there we have to kind of play with the visualization a bit. But then what we did from this basically in panel B is trying to break things down into the different areas. And so she basically tried to manually sort here the brain areas in terms of like cortical, frontal cortical areas visualized at the top of these graphs. And then somewhere you have primary visual cortex in the middle of the graph, and then hippocampus, LGN, striatum basically being towards the end. And I think this would be a nice way to illustrate the number of units that you get. Yes, Jerome?
I'm just wondering if it's worth comparing with the SST opto-tagging as a sanity control. Would you expect SST to be one of the class wave map detect?
Honestly, I don't know whether they have a very stereotypic waveform. I wish they had. If they had, then I'm wondering why.
Yeah.
Why would we not use physiological properties to identify them? I'm not sure. I think it would be good to check. I actually don't know.
One thing that is worth looking into also is we designed the zebra stimulus to cluster the inhibitory neurons because the VIP SST and PV cells have very different spatial temporal orientation tuning properties, and so they should cluster separately.
We can check.
You could check. Yep.
Yep.
During the zebra stimulus in particular.
Yeah, that sounds good. Yeah, I think here Severin, yes?
I'm curious about panel A with the one that is high firing. Is that like a PV type cells or like narrow spikes?
I have to check I think it's class 25. I'm not sure if that's visualized here. No, not. I have to go back and check what the waveform looks like.
Okay.
Yeah. Yeah.
There's a beautiful dynamic figures to make. I'm just mentioning, but
yeah, that's clear. That's missing. This is very old school, but at the same time, it's loading all experiments and preloading them on a Google Drive, so that one doesn't have to stream each time, because that's the painful part of this type of effort when you try to do all of the animals, basically.
Yes.
But so that one we actually figured out. That might actually also be a nice integration into the code, because really storing this on a Google Drive might be actually something that makes this quite feasible for a lot of people, instead of having to stream all the time or having a laptop that allows you to save this data locally. That's another, I think, bottleneck basically for people.
So what did you put in the Google Drive? The entire data set, or?
So I have to double check with Mariana, but I think she downloaded it. She downloaded once all the files and then saved locally the metrics of the good units, so that she could go back into all the different metrics of the units.
But how big is this file?
I have to double check. I don't know.
Yeah.
Yeah.
Sometimes some of us can be committed to the repo, but depending on how large it is.
Yeah. Okay. Yeah, and the last thing she has done is basically then we tried to look at the proportion of cells we could, because we were trying to think, right? I mean, some of these cell types should be probably enriched in different brain areas. And I think what's nice in panel C is that the LGN stands out as being very enriched for particular waveforms compared to others, right? In terms of like very overproportional blue representation. But I think some things like that could at least help a reader decide, right, whether you have enough units. I would actually also like to see the spread across individuals to know whether have enough units on average, right, across the mice. And then how these different cell types are presented in these areas. And I thought parts of this could be basically panel E and panel I in terms of quality control of the EFIS, irrespective of oddball responses or any specific context, basically.
I do want to mention that the developer of WebMap, Kenji, is he used to work in the Office team. So I feel like you could very well send your slides to him and have a conversation just to make sure that it's pushed to the limit of what it can do.
Sounds good. Yeah.
I'm happy to connect if that's helpful.
Okay, that sounds great. Yeah.
Okay, Samuel?
I was just wondering, actually, with this discussion and the SNR discussion beforehand, if one possibility is that the neurons in the brain or the bits of their things that are most sensitive to the conditions that are of interest for a particular oddball, for example, they're also rare and of particular subtypes, let's say. How does one try to make sure that the kind of, because a lot of these kind of thresholds or sanity checks, quality checks may be dominated by a large number of neurons that are, say, irrelevant for the computation at hand. So how do you make sure that you don't accidentally reject the kinds of neural signatures that might actually be most important for doing the computation? I'm just curious there, because I feel like what you just presented, Sarah, is like some really nice, you could get really stringent on your criteria, perhaps, but perhaps the neural elements that are actually providing the right signals are also more variable in, for example, how they're responding.
Yeah, I think you have to go both ways, honestly. I think you have to first look for oddball sensitive neurons and then maybe check what are the properties of these cells ultimately. But for the data release paper, I was now thinking just the other way around. And what would I want to know to convince myself that this is a good data set? And then I would like to know the signal-to-noise distribution, even if in the end the oddball sensitive neurons might be signal-to-noise 2.5. I don't know, but at least it would give me confidence that this is a good recording. And for some aspects, for instance, if I'm interested in population decoding, I would need to know how many neurons do I have in a given area, how reliable is this across mice, so I can get a sense of can I actually do the analysis I'm interested in. But yeah, honestly, I would use the tool like what David and Roberto showed to first find the interesting neurons and then go backwards, right? And then identify all these SST cells, where are they? But for quality control, I would go the other way around.
Stefan?
For quality control, I would actually say that it would be much better to not do an analysis of interesting things before filtering things for quality control. Most definitely, mainly because a lot of statistical measures are not really good for that. And we might be fooled by picking things which would have a spurious oddball-like effect due to long tail distribution, low signal to noise, or things like this. So my strong recommendation, if as a statistician and mathematician who's listening to this, my strong recommendation would be to use a very strict ordering of quality control first, data analysis later, no matter what, because otherwise you might get fooled and might be tempted to do other things.
I think this is a great paragraph contribution to the manuscript to make, Stefan.
Sounds good. I'll be happy to do it.
I can point you to the way.
Right. So shoot me an email to follow up where we sort this. Yeah.
So it's 9:59. So I want to thank all of the 4 people that prepared. This was fantastic. I feel like this format works well. So let's continue next week. Antonio?
Yeah, I also prepared something, but I forgot to post it on the discussion. But I will be happy to present next week if possible.
Yes. I also encourage anybody doing analysis on the 2-photon data to post, because we want to keep all the modality progress. So if anyone wants to present 2-photon, you're welcome. So we'll do the same next week. People can post ahead of time on the discussion thread. I'll put the meeting on the website, and then we'll follow the same process. Thanks, everyone.
Okay, thank you. Bye-bye.